To bio class. Today we'll discuss biomacromolecules. Biomacromolecules. Before starting the biomacromolecules, here is a short para is there, that is primary and the secondary metabolites. Okay, so this part, primary and the secondary metabolites. This part. Okay. Now, what is this? What is called metabolites? Metabolites, you can say that any substance which is necessary for the metabolism. Any substance can formed or it is necessary. In one word, you can say that it is ingredients. Ingredients of the metabolism. All right. So metabolism here, I mean the process of metabolism, the very big term it is, that afterward it is discussed. For that, these metabolites are required. It can be, I mean, raw materials, or it can forms also. Both type of meaning is there. Here it has a clear division of the metabolites, which is in primary metabolites. Primary metabolites, you can see that here in the figure 9.1, where the elements, just recall that the hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, all this. And here the secondary metabolites. Secondary metabolites, here is a list, you can see that. In this list here, the pigments, alkaloid, essential oils, toxins, lecithin, drugs, all this you can see the secondary metabolites. The morphine, here it is the list. You have to little bit prepare this part. Okay. Now, next part we can move that biomacromolecules. So biomacromolecule, we have to understand that at first beginning of the chapter, the one experiment we did with a trichloroacetic acid, and we have got one acid soluble substances, and I mean acid soluble and acid insoluble fraction. Here it will be insoluble fraction will be macromolecule. Let's see their molecular weight here 18 to 800 Dalton, so much bigger in size. The macromolecules, which are the weight is less than 1000 Dalton and usually referred to as biomolecules, and where it is macromolecule is much bigger, and it is acid insoluble fraction. So in biomacromolecule, it is there that the protein, polysaccharides, nucleic acids, these are. So let's discuss one by one that biomacromolecules. At first, come here protein. Protein are polypeptide. Already all different types of amino acids, 21 or 20 different type of amino acids it is discussed and told. We can see once again of it, that one, that is you can see the long list of the amino acid. That characteristics, I think you know that one amino group will be there and one carboxyl group will be there in every, at least one, it can be more than one, and different I mean heterocyclic and all other it is there. The basic acetic, this part already it is discussed. Now protein, I mean amino acid combines into dipeptide, polypeptide, peptone, and ultimately it turns into protein. So let's know it's about the bond little bit. So here it is. So this is one amino acid. You can see that the carboxyl group and this amino group, this amino acid 1, and here amino acid. Here in each 2 we have written in that way, and another amino group. Now in bond between these two, HO, I mean it is water, and it forms that peptide bond, COHN. So this, in this way, one amino acid, it connected to the another, it formed the dipeptide. In that way, the tripeptide, polypeptide, peptone, and I mean protein it is. Okay. So see this one here also the same thing. It is there in another graphical pattern. You can see this one. This you can see that this water molecule, it moves out and show that here peptide bond, in this peptide bond forms, and it forms the dipeptide, and water it is removed. So again if we do hydrolyze, then it will breaks into amino acid. So reverse, if we go, when we take the protein as a food, then in the process of digestion it is broken down, and at last the amino acid it enters inside our body. Alright, about amino acid, so from the long list that we have to understand that all the amino acids are, we cannot synthesize in our body. So there are few. See that essential amino acid not synthesized in our body, and we have to take from the food. So these are the essential amino acids: valine, leucine, isoleucine, methionine, threonine, arginine, lysine, phenylalanine, histidine, tryptophan. These amino acids are called essential amino acids. Alright. So next we can see this the polysaccharides. So polysaccharides is next macromolecule. Polysaccharide, to understand the polysaccharide, that you have to understand this part here. Look at this areas. Look at here. This is the carbohydrate. You can divide monosaccharide, disaccharide, and the polysaccharide. Okay. Monosaccharide having a single type of structure, which is very common is glucose. So here is a common formula, CHO and whole N. Here N, if you put 6, then that will be the hexose sugar, which is glucose in aldose form, and ketose form fructose, which is there abundantly in our body and organism's body. Now in DNA, which is available, that is pentose sugar, which is ribose and the ribulose, this where the value of N equal to 5. So one monosaccharide, these are monosaccharides. Suppose glucose is a monosaccharide. So when this glucose will join with glycosidic bond, so this is one glucose, this is another glucose, will join, it forms the disaccharide. But when many monosaccharide joins, many are there, at 40,000 or more, so it turns into polysaccharide. So polysaccharide, simply from that we can able to understand that it combines of many monosaccharides. Almost like before that we learned that amino acid, I mean protein, it is just that chain of amino acid. Here the polysaccharide, it is just chain of the glucose, mostly glucose. Okay. So let's know about this the different types and the part of it. Now in polysaccharide is formed and the joining of monosaccharide like glucose. So how glucose is joined? The first come here, the glycosidic bond. Here it is that it is glucose. This is alpha D and beta D. I'll tell you afterward. First see this. This is the glucose. Here we have drawn in that way. Here OH and here also that OH. I think you can see that yellow, that patch, this part HOH. It means one water molecule. It comes out, and this O, it comes here, and it forms the bond. That is called the glycosidic bond. All right. Same way that when we will take the polysaccharide, because in nature the polysaccharide is available like starch. When we take the starch, then the enzyme with the help of water it breaks and makes it into the glucose molecule. That glucose it enters inside our body for the energy. Okay, so this part in this way this bond will form. Now alpha D and the beta D, just know that in chemistry I think you will learn better. Here the structural little bit difference. The alpha D glucose, the carbon number 1, here the place of the hydrogen and hydroxyl, this 2, and in beta D the N carbon 1, placings of the hydroxyl and hydrogen is just opposite. So these are the difference. Okay, now here in your book you can see the cellulose, glycogen, the diagram also there, and the how it can form, how it looks like. Let's take a look. Now about the glycosidic linkage, one thing you have to know, when it goes in straight chain, and this linkage it is 1 with the 4, 1 with the 4 carbon, so that it is called alpha 1,4 linkage. But when it comes in branch, see that here the sixth carbon it joins with the 1, so it is called the 1 and 6, 1 to 6 linkage. So 1 to 6 linkage, it indicates that it is branched chain. Now starch, which is the most abundant in the nature, it consists of the two components, amylose and amylopectin, 1:3 ratio. What is amylose? Amylose, it contains only that 1,4 linkage. So it can be number is used as 200 to 2000. It can be a coil like this, but it is straight. It is no branch is there. It is straight chain. But if we see that amylopectin here, amylopectin here it is, the straight is there, it means the 1,4 linkage is there, and in this only this areas where the branch will start, that is 1 and 6, alpha 1,6 linkage also there. Okay, so this type of the branching structure also there. It can be 2000 to 2 lakh glucose units is possible. So potato, banana, wheat, see that this type of structure it can be possible. Here is line, it means that this chain, this line, it means this the chain. So glycogen is another one which is found in animal. Here also that both the types are available, 1,4 and 1,6, the glycosidic linkage, and it looks like this type of branch. Here is 30,000 to 40 lakh glucose molecule can be possible. Now next one is cellulose. Cellulose, it is beta linkage is available. See that this beta linkage, beta 1,4 linkage, beta D glucose, here it is there. Here also the huge unit is there, and 1,4 link, it is in straight chain, it is there. This found in cellulose, you know that it is plant cell wall, it is there. Okay. So and one thing, interesting fact, that the human do not have the enzyme to break the beta linkage, and so that we cannot digest cellulose. We don't have cellulase enzyme to digest it. Okay. So this part is only you have to study. Okay. Thank you.